Hello, listeners. Yamina here. Welcome to the Dr. GPCR podcast. Before we dive into this episode, I have a few announcements to make. We are working on a new program geared towards transitioning from academia to industry. Powered by Dr. GPCR, our new program is called Life Career Job. The goal is to support you in your efforts to find your next job in industry, plan out your career trajectory, create a life, and support the lifestyle that works for you. If you'd like to be notified when this program becomes available, fill out our short survey at lifecareerjob.com. This new ecosystem will also include a new podcast that will be launching in September 2021. Our guests will be scientists, recruiters, and hiring managers, and everything in between. We want to help Ph.Ds find their ideal career path and inspire you with real-life stories. The second edition of the Dr. GPCR Summit will be held from September 13 to 19, 2021. We are planning a combination of live talks, pre-recorded talks, and live workshops. We have a great lineup of live talk speakers, including Dr. Brian Roth, Dr. Patrick Sexton, and Dr. Jürgen Weiss. Also, we are happy to announce that we'll have a live Q&A session with Dr. Bob Lefkowitz and Dr. Randy Hall to talk about GPCRs, science, and Dr. Lefkowitz's memoir. Visit drgpcr.com/summit today to see the full program. We're also launching our GPCR consulting services. You'll soon be able to find the profiles of our carefully selected consultants on our website at drgpcr.com/consulting. You'll be able to find the help you need for your company. Stay tuned. Are you subscribed to our YouTube channel? If not, please subscribe today. It's not only a great way to catch up on our recorded events, such as the Dr. GPCR Virtual Cafe, but it's also a great way to support Dr. GPCR. And now, let's dive into this episode. Hello, everyone. This is Yamina from Dr. GPCR. I am very, very happy to have Dr. Sudha Shenoy with me today from Duke University. Hi, Sudha. How are you?
I'm doing all right. And how are you?
I'm great. I'm excited. The weather is getting more and more beautiful, and I'm very happy to have you with me today.
I'm so excited as well to participate in this podcast. And, you know, the weather is not so bright here. It's kind of rainy and gloomy, but it's okay. It's okay.
We sent you some of that rain. We've had rain for so many days lately. And
you did. We'll send it right back.
Thank you.
Okay.
Thank you. We'll take it. It's the Boston area, so I think we can get by with that. So I'm glad that we're doing this today, and we finally got to chat. Would you please introduce yourself to our audience?
Okay, as you mentioned.
Although before you do that, I don't think you need an introduction, but please proceed.
I do. So as you mentioned, I'm at Duke University. I'm a associate professor in the Department of Medicine and Cell Biology here at Duke. And even though I'm in the Department of Medicine, I'm a Ph.D. scientist, and I'm actually affiliated with a center called Cardiovascular Research Center, where many basic scientists are housed. And our lab research is focused on understanding the roles of ubiquitination, post-translational modification in G protein coupled receptor signal transduction and intracellular trafficking. So that is our main area of research.
That is so interesting. I think ubiquitination is also a very interesting mechanism to regulate GPCR function.
Yes.
That's interesting. So how did you get into studying GPCRs?
Well, I can tell you a little bit about my path to science and scientists. So even as a kid, I was always interested in biology and chemistry, even in high school, and But then my real sort of attraction or a drive that you can say about doing basic research came during my PhD training. So I did my graduate studies in the Department of Biochemistry at Oklahoma State University. And what I would do was I would be waiting for my lecture classes to be over because I want to get to the bench and I want to set up my experiment. And somehow I had developed an attachment or a longing to do these experiments and then get them done and then see whether I got any result. And then I wanted to interpret those results. So I was very, very enthusiastic about doing experiments, even though my first 2 years in my PhD program was full of dead-end experiments. It's not because the techniques didn't work. I was pretty okay. I learned the techniques very fast. But the problem was because my project was kind of a challenging one, if I can say that. So several students before me, and I didn't know this when I undertook the project, but several students had left because they couldn't take on the stress because nothing would move. And this is because the project was to identify and clone a small transmembrane protein called QPS3. Now, this protein is expressed in the mitochondrial inner membrane, and it's one of the subunits of the complex 2 called succinate quinone reductase. However, this protein was very elusive to purify or clone, and many labs had given up because they thought this was just a ghost band of another protein called QPS1 or QPS2, because you couldn't see that very well in gels. So they always assigned this to be like a contaminant or a ghost band and things like that. My advisor was bent on saying that, okay, we should find this. This is something that we have to do. So after many months of struggling with the traditional approaches that we had, what I did was suddenly I thought, okay, why not I completely denature this complex and then run it on the gel? Yeah. And then rather than using the traditional methods, I added different amounts of urea. And then I split the complex, I ran it through a column, and I then ran it on a gel. And suddenly my band was so bright, and I could cut it out of the gels and purify enough of it. And this method just allowed me to get the protein sequence of this protein and the amino acid sequence of this protein. And then I finally got this clone out, and that was my first paper in JBC as a graduate student. It turned out to be a very hydrophobic protein with only 100 amino acids. It's got 3 transmembranes. And so this work, finding this, was really helpful. I could do the follow-up work. I sort of mapped the quinone binding sites, which was another aspect of my PhD work. So it led to 2 first author papers at that time. And this sort of experience, I don't know what it did to me. And once I got the taste of this, I was kind of hooked. And I was very passionate and very curious to discover and learn more about sort of biochemical mechanisms. And this is what led me to look for postdocs. In a different area. So I wanted to move away from cellular bioenergetics area. And then I went to, I started looking for labs where I could sort of do something more which tells me about the biochemical pathways in cells, and G protein coupled receptor signaling was at the top of my list. And this was because of course, reading through a coursework. There were a couple of lectures on G proteins when I was a graduate student. And I was really fortunate to get an opportunity to work for Dr. Lefkowitz. And I joined his lab in 1999. And this gave me an entry to the GPCR community. So, but when I joined Dr. Lefkowitz's lab, I did not work on a receptor. So my assignment was to find a function for a protein that had just come out of the Yeast 2 hybrid screen, that they were doing at the time in the lab for beta-arrestins. And this protein, you may know, this is MDM2. Mm-hmm. And so I was assigned with finding a functional role for MDM2. And what did I do initially? I looked at it from a different angle. I said, okay, MDM2 is known for its role in P53 regulation and cell cycle and apoptosis. So I did many months of work looking at those aspects, but none of them were kind of in line with what was going on in the lab. Everybody was doing GPCR pharmacology or signal transduction. So I was always feeling left out, and I wanted to do something that connects MDM2 to either GPCRs or arrestins. And one of the qualities I had was because I was coming into a new lab, a new field, I knew nothing about it, and I would always make some time to read. I couldn't read full papers, but I would at least read the abstract of as many papers as I can whenever I had time. So as I was reading and going through the PubMed, I found papers that had just come out that mentioning that MDM2 is the E3 ubiquitin ligase that had just come in a year or so, or 6 months before I joined. And I saw that. And then I also found, okay, there is ubiquitination. Let me see whether there is any ubiquitination of arrestins and GPCRs. So there was some, I think there were some papers on arrestin, I think the visual arrestin being degraded, but it wasn't very clear. But I remember that what really helped me to get into this field is the papers on the yeast GPCR, sterile 2 and sterile 3, where they showed that ubiquitination occurs on those receptors, and that helped the receptor to internalize. And once I saw that, I said, okay, let me figure out whether there are any mammalian receptors that are ubiquitinated. There were none. There was nothing. So I said, okay, let me look at some receptors. And I discussed with Bob, and he said he really liked the idea. And then I went around literally to everybody around Crohn lab, even in Bob's lab, looking for flag-tagged receptor constructs. So I collected a bunch of them because, the first experiment you want to do is a pull-down assay. You need a tag. We don't have ways to pull down different receptors. We always tag them. So I collected a bunch of different receptors that had flag tag. And so at that time, ubiquitination, mostly links proteins to degradation. So which receptor has the shortest half-life? And reading the literature, I know thrombin receptor was one of the best candidates because it has the shortest half-life. It doesn't recycle. It's just one-way traffic. So I had a flag-tagged receptor. And then what would be my negative control? That's GPCR that should never be ubiquitinated because it should be so stable. And from my discussion with people in Bob's lab, at that time, I think there was a group purifying the beta-2 receptor. And they told me, the beta-2 receptor, we leave it on the bench for many hours. It's very stable. And if you look at the papers published, if you want to see degradation, you have to wait for 1 day in cells, like 12 to 14 hours to see 50% reduction in the receptor. So that should be, okay. Then I said, okay, this receptor must be the best negative control. So I did my experiment. Of course, I didn't get expression of many of the constructs, but I forgot something. So what was, upon the suggestion from my mentor, Bob said, you're doing all this, you probably should also look at sterile 2. And I said, okay, I'll look at sterile 2, but then we got a construct, and I moved it into a mammalian expression vector, and we had the alpha factor synthesized because Bob said, whatever you do, you have to do the minus plus agonist. So I had to collect all the agonists. So we did minus plus agonists. And of course, many of the constructs didn't express, but I was able to see some ubiquitination for some of the receptors. And for sterile 2, I could see very nice mono ubiquitin band. But what was striking was the only receptor in that experiment that showed agonist effect was the beta receptor.
Of course.
None of the others showed any agonist-induced ubiquitination, and that was it. And one thing led to another, and then I was a pretty I don't know, my attachment to bench might have helped, or my curiosity and drive might have helped, but I think within a period of 2 years, I think we had that paper in Science 2 years into my postdoc, but it was round the clock working. It was no time for nothing, but it's not round the clock. I still had time. As a mom, I went back home, cooked for my family. I came back, but I was very, very, time bound. So I would plan things, I would do things, and I was very strict about my timing stuff. So I got everything done, and we had this nice paper come out. So this is my story of getting into the GPCR field.
That's fantastic. I love the stories, and I love how much details you provided us. And I want to take you a little bit back to your PhD and maybe ask you, how did you choose your PhD lab? And I have a follow-up question to the PhD portion of your career.
So interesting. So basically, I completed my master's in India. And I think after that, after a couple of years, my family moved to United States, and we arrived in Stillwater because of family reasons. Mm-hmm. And I started looking for, I always wanted to do my PhD in biochemistry, so I was applying to the nearby schools, and I was also taking sort of courses. Mm-hmm. And when I took my biochemistry course, I came to know my PhD advisors, and I went and talked to them, and I worked in their lab for a year or so as a research assistant, and then I started my PhD. So that's how I got into the lab where I did my PhD.
That's fantastic. You're making a great point to going and doing the research and finding out what you like and what you don't like. And I think that year that you just mentioned, working as a research assistant in the lab, really helped you realize that you love working in the lab and that was also the right lab for you. And I think that's very important to note.
Yeah, I did. I learned a lot from my PhD training. And of course, it was all on protein purification, protein biochemistry, but that has sort of helped me to do a lot of things later on, even when I was working in other aspects in mammalian cells. It has really helped me to gain some fundamental knowledge about all these biochemical principles that we use, in no matter in what you do. You need to have some chemistry background, I feel.
I think so too. Where did the idea of denaturating the protein and using urea come from to solve the purification part of your project?
Yeah, so that's a very interesting question. So one of the things if you think about complex 2, you have to purify it and in very carefully, and you cannot expose it to oxygen because it will denature the protein. You have to purify it in a certain way and under anaerobic conditions if you want to have a pure enzyme. So you always end up getting very SQR with very high activity. So my lab was purifying this from actually beef heart. So that was their model, beef heart mitochondria. And honestly, I was trying various conditions. First, I was trying to increase the activity of the complex, and I thought maybe that can help for me to get a better band on gels. But we would have very active preparations, but it would still be like a ghost band. We use different types of gels, and I said, okay, maybe I have to split the hydrophilic and the hydrophobic subunits, into two components. And that was actually done by some groups using urea. So they had actually split some of the hydrophobic components using a urea column. So maybe I got some clue from that, but then I did not expect that would help me to purify this subunit at all. I did not anticipate that. But in a way, I think I ran away. I ran in the opposite direction and took sort of a shot in the dark, if you can call that. Yep. I tried everything else except for denaturing the protein. And I felt, okay, I'll denature the protein and then renature it. And you can do it. If you can denature and renature, if you can remove the urea by, buffer replacing or dialysis, you can renature to a certain extent, but you cannot put it all together. But my purpose was not finding activity, but I wanted to sort of be able to get enough protein to cut it out of gels. If I cannot see the protein, I won't be able to cut it out. And that was the hurdle or the roadblock at that time.
That's fantastic. It sounds like it was a calculated risk that you needed to take.
Definitely. It was a calculated risk. This happened so, I feel it's so long ago, 1990s. I'm trying to remember exactly what happened, but if I had access to my notes, I will know, because I'm very good at keeping notes of my daily notes. I would write, even if I had to go somewhere that day, I will write it in my notebook.
That's an incredible.
I keep my lab notebook like a diary.
That's
so
perfect.
It always helps me to figure out what I did on that day. So I wish I had it. I could tell you more.
I think for the events having happened in the 1990s, I think you seem to really remember everything that happened, and I find it phenomenal that you sat down, you read the papers, you looked around, and you were able to zoom out of that problem that you were having and found a solution that ended up working, which is phenomenal.
Thank you. I think I was probably lucky. I mean, I don't consider myself lucky in a lot of aspects, but in some aspects, I think luck has helped me. Yeah. Or you can call it hard work. I don't know.
I think sometimes it can be the combination of both. I think one brings the other, probably.
It can be. It can be. Yeah, I think so too. But later on, I think as I moved on into Bob's lab, I think a lot of people have helped. I mean, colleagues, especially not only in Bob's lab, is extended family. I take advice, help from a lot of people. Michelle Bouvier, Jeff Benowik, Brian Kobilka, from time to time, they have really helped me either with reagents or they are very generous. Yeah,
that's fantastic. So you completed your postdoc, and then you actually were able to.
So basically, we were. I talked about how I got into looking at receptor ubiquitination, but but at the same time we were also looking at beta-arrestin ubiquitination, and as you may have seen in that first paper, we were able to show that ubiquitination of the receptor and that of arrestin have a distinct, and they had distinct functional effects. especially for the receptor, it looked like it was mediating probably lysosomal trafficking and degradation. And then for the beta-arrestin, we showed that it was probably important for mediating receptor internalization. And of course, there are follow-up papers that showed that beta-arrestin ubiquitination is like a mechanism that helps it to bind very stably to the receptor, but also to non-receptor proteins and things like that. So towards the end of 2005, I was able to get a couple of grants. One is a career award from the AHA, and I also got my first R01 in 2005. And I started my own lab at Duke, here at Duke. I was recruited into the position, assistant professor position, into the Department of Medicine and Cardiology at that time, and also cell biology. So since 2005, I am sort of an independent lab here at Duke, but it was not perceived as independent. People, whenever I wrote my grants, people would always say that, oh no, you're in Bob's lab. So it took many years after that to figure that out, that I had an independent group, and a lot of people are here. We sort of we train with Bob and we remain in the same place. And in the GPCR field, I think everybody knows, but I'm saying if you in the outside field, for example, study sections, they always don't like when a trainee stays back in the same place. And I think sometimes it's good, but sometimes it's kind of a hurdle that everybody has to cross at any point. If you happen to stay in the same place as your mentor.
Yeah, but then again, when you have Bob Lefkowitz as your mentor, it's not always a bad thing.
No, it's not. It's not. It's not.
Which is amazing. So you took the reins of the ubiquitination of GPCRs and of arrestins that you started out in Bob's lab and established your own lab. What are you working on today?
Well, actually, pretty much the same. So I began my studies characterizing ubiquitination of the beta-2AR, and which is one of the most studied GPCRs. So in a way, I'm happy that my initial screen helped me to focus on this receptor, because you have a lot of tools, a wealth of information. You have Ray plasmids. You have various hydrophilic, hydrophobic ligands. You have protocols to study it. So it made an attractive receptor for studying. So, but on the other hand, and besides this, the beta receptors are indeed important targets for treating heart failure. So they are not like it's an important area of research. However, as I just mentioned, it was getting very, very difficult to get more and more funding, because I always had this criticism from my studies, from reviewers, that a focus on beta receptors has no novelty. It kind of overlaps with my mentor's research area. There's nothing left to understand about these receptors, because there are already thousands of papers out there. And if I think back, as hard as I have tried, Yamina, to move away from the beta-2 receptors, something or the other has pulled me back into studying the beta receptors. For example, suddenly we detected, I don't know why we even did those experiments, we found that the beta blocker carvedilol can also induce ubiquitination of the receptor. And it was not a result that you could just say, okay, leave it alone. You had to pursue it, because it was so interesting. And one thing led to the other, and we found that it is regulated by a different mechanism than what happens when you stimulate the receptor with agonist. So when you stimulate the beta-2 receptor with an agonist, there is a particular E3 ubiquitin ligase that ubiquitinates the receptor called NED4. Beta-arrestin 2 is definitely required in that process. It functions as a ubiquitin ligase adapter. But on the other hand, for the carvedilol, it's not mediated by NED4, doesn't require beta-arrestin, and it's mediated by a totally different E3 ubiquitin ligase called MARCH2. And so here you go. Again, we are into the beta-2 receptor system. And then we also had this situation where we were looking for substrates of deubiquitinase. And at that time, my postdoc tried different proteins outside the realm of a receptor protein or even the beta-2 receptor protein. But basically, most of the assays worked so beautifully with the beta-2 receptor when we tried. And there you go. We showed that USP20 and USP33 regulate the recycling of the beta-2 receptor. And again, we were into the beta-2 receptor for no reason, even though we, at the beginning of that whole project, we tried some other receptor and nothing was moving forward. And then, just as a control, my postdoc tried the beta-2 receptor and started working. So again, we had that. And then we were looking at USP20 protein, and it would always migrate as a different band in agonist stimulated, in iso stimulated samples. And when we pursued that, we found out it's because of a change in phosphorylation that correlates with the mobility. And there you go, you have receptor-induced phosphorylation of USP20, again, the beta-2 receptor. So we always had something new and interesting to come back to the beta-2 receptor. And even to this day now, we just had published a year or so back, we showed that this USP20 regulates both the beta-1 and the beta-2 receptors. But however, they have, there are very key differences. For example, with the beta-1, only the phosphorylated form of this deubiquitinase USP20 associates very strongly. It has a strong interaction. Whereas with the beta-2, only the dephosphorylated form of this deubiquitinase has a strong interaction. So again, I feel this is a very interesting and very novel area to pursue further, enough to motivate me to make knock-in mice. So now we have knock-in mice made with changes in USP20 just for that residue. So serine to alanine and serine to aspartame. So we'll see what happens when we get to characterize. Mouse work is kind of, it has its own ups and downs. So sometime in the near future, probably we'll find the difference as to how it is regulating the beta-1 and beta-2 subtypes in the heart. So that's what I'm looking forward to in terms of what are the new things, even though same receptor, but I think there will be some new information, I'm hoping. But I'm still trying to find new GPCRs to like, if I can say that.
So we can definitely say that it's the beta-adrenergic receptor as your favorite GPCR.
I cannot. It is a good receptor. It's a good system. And I'm very intrigued by the new information that we are getting. But, to a certain extent, I do agree with the reviewers. It has been studied so much. So it is one of my favorite receptors. It's not the favorite, but it's one of my favorite receptors. So basically what we have tried to do is, you can take a receptor and look for either ubiquitin ligases or the deubiquitinases that regulate this receptor. And, there are hundreds of ubiquitin ligases. There are about 100 DUBs. So you can focus on one receptor rather than doing that, because we have developed tools for studying a handful of DUBs, which we think are regulating several GPCRs. So one of the goals is to take these dubs and go in the reverse to see how many GPCRs are regulated by each dub in any system, and by doing some binding assays or even ubiquitin assays. So that's something that can lead to a new direction if you got even something novel that we didn't know about. So we are starting from the reverse, and we'll have to wait and see which GPCR pathway this sort of an approach is going to lead us to. So that's something that I'm looking forward to. I don't know when. And we are also under, one of the interests is to pursue what is the role of ubiquitination in terms of beta-arrestin functions and in terms of in signaling. Because I personally, if you look at all the work that is published out there, it's very clear that site-specific ubiquitin on beta-arrestin can itself form a code. You can call it a ubiquitin code, but how this code fits into the context of bias signaling, it's a tantalizing question. We don't know, but it's sort of a question that I always like to ask myself, and I always try to find a way to pursue this angle. And the other interesting aspect of ubiquitination is that even though for many receptors, we think about receptor binding to beta-arrestin, you have the G protein coupling, and then you have phosphorylation, and then you have beta-arrestin binding. And beta-arrestin mediates internalization. But it is possible that there are some receptors which can sort of bypass the beta-arrestin dependence and totally depend on ubiquitin for internalization. It is possible, and for trafficking. And there is a hint of mint in a recent work that we did with Mark Cron's lab. And, they were looking at a particular motif which sort of induces bias, either beta-arrestin bias or G protein bias with the ghrelin receptor. And then using the same principle, they also made the same mutant in a chemokine receptor called CCR1. But you couldn't see the difference. Both when the wild type and the mutant, they were recruiting arrestin in the same way. On the other hand, when we looked at ubiquitination, it was different. And the localization of the receptor was also different between the wild type and the mutant. So ubiquitin itself can function either in a way that it intersects with other transducers, or it could be itself a signal for receptor to localize to different compartments, at least for some receptors. And I think the CCR1 could be the first example. We don't know. There could be others out there. So GPCR trafficking regulated by ubiquitination and deubiquitination can have roles that overlap or intersect with beta-arrestins, but it can also be an independent mechanism in itself. Okay, so
I love it, because if we go back to the beginning of my question, where you were mentioning that depending on which beta-adrenergic ligand you were using, you were detecting a different effect in ubiquitination, and ubiquitinase was different as well. I think that in itself is just magnificent, because you're showing bias, and it's not your typical beta-arrestin G protein bias; it's a ubiquitin bias.
Exactly.
Which, yes, again, I love that, because then yes, people can tell you, oh, it's beta-adrenergic. It's a great receptor system, a model receptor, but then it can show the subtleties of GPCR function. And I think that is key, and that is just beautiful work.
Thank you.
I love that. I was listening to you talking, and I kept in my mind, oh my God, this is mind blowing, that the regular bias is usually that people talk about is beta-arrestin and G proteins, but there are other components in the cells that will dissect that functional response, which I think is just amazing.
Yeah. So I mean that's where I'm. All my thoughts are actually naturally.
What type of assays do you typically use to assess, ubiquitination?
So this is something that I always delve on because so the standard assay is kind of cumbersome. You have to do immunoprecipitation of either the ubiquitin or your protein, and you have to run Western blots. We For some systems you can use FRET and BRET, but the problem with that is it may not be that productive or that useful when you are trying to track polyubiquitination, because the polyubiquitin moieties actually start to quench your FRET signals. And I have spent years trying to develop FRET assays, and those are all failed experiments in my notebook. And probably those years when I had very little to publish and got criticized of low productivity also from the reviewers. So I have tried, but there could be receptors where monoubiquitin is sufficient to see an effect, and for those receptors, probably BRET or FRET could be a good assay. I mean, it could be developed into a throughput assay. Recently, we were able to show that you can use BRET for detecting sumoylation, the effect of beta-arrestin sumo. And in that paper, we also compared the beta-arrestin ubiquitin chimera. And you could see the difference in using the standard confocal assays. You can see that the beta-arrestin ubiquitin binds the GPCR tightly. But if you take the same constructs into the BRET assay, now you can see that no, it's even worse than the wild type arrestin. It doesn't bind the receptor. But on the other hand, the sumo-elated beta-arrestin sort of helped us to move away from this technical aspect. And it does show a stronger association with several receptors that we checked. So I think if there are receptors where you don't have to worry about the polyubiquitin chain forming, and you are able to get an agonist effect, and I think that's something that would be really great. Really, it would be really useful also. Yeah.
That's amazing. Amazing. So when do you expect to get any mouse results, mouse data?
We have the mouse made. We are now, sort of back crossing. We have some preliminary data. So it's ongoing. It's ongoing. Yeah.
I'm excited to see that. But I feel like, working with mice, the mouse experiments have their own timeline. They live in their own realm. And it's not an easy task.
Yeah, I'm very interested in understanding how the beta-1 and beta-2 receptors are sort of dysregulated in heart failure. So, the differences that I mentioned characterizing ubiquitination and deubiquitination, especially the way they interact with the DUB, seems to be one interesting difference in terms of regulating the two subtypes. So I think, but I don't know. We'll have to wait and see what the receptor levels are in the left ventricle and things like that. And that's still ways off. But I can still, I'm still very excited even thinking about those experiments on, future experiments.
Did you ever see any differences between ubiquitination of beta-arrestin 1 versus beta-arrestin 2? And does it have any effect on receptor function?
A very, very good question. So most of the effects that you see with beta-arrestin 2 are also reproducible with beta-arrestin 1. But the magnitude of the effect is not very high. So we do have a beta-arrestin, unpublished, of course, a beta-arrestin 1 ubiquitin chimera that, we probably tested it. It functions in the same way as porostin too, but we haven't really tested these constructs with a wide range of receptors. So we have always been sort of limited by the number of receptor systems that we can handle and test. So I think there could be differences if we are able to get our hands on more receptor systems. We would probably be able to see a difference between beta-arrestin 1 ubiquitination and beta-arrestin 2 ubiquitination. Yeah. But recently, we have become very interested in a second family of GPCRs, the glucagon family receptors. I think you may have seen that we had a recent paper in JBC where we reported that the glucagon receptor, which is one of the receptors in that family, it presents a sort of a different ubiquitination profile. And I think it's very interesting. And we also have some unpublished data suggesting that this difference could have a real role in signal transduction and physiology of this GPCR. So that's an interesting angle that's at a very beginning stage at this moment. So we'll see what happens, and we want to extend those findings into the other family members, which are of course are well studied, the GLP-1 receptor. Yeah. So we'll have to wait and see how that goes. So those are, I don't know if it will allow me to take the ubiquitination project into a different family of receptors. And, I still have to work on the beta receptor. I mean, I'm really bent on finding out what happens to the beta 1 and the beta 2 in the heart. We'll see.
That's really exciting, and you're right. When we started chatting, and you mentioned beta-adrenergic receptors, most people think, as reviewers, it's been done. We know everything about it, and that's not the case.
Yeah, but one of the things that I would like to mention is, of course, we have a lot of ways to target the receptors, and we use beta blockers that have been in clinic. But you have to remember, all these drugs, they were generated at a time when we did not understand the biology of the receptors so much. I mean, there was no biosignaling, and nobody knew anything about arrestin biology, arrestin signaling, or ubiquitination for that matter. And regulation by dubs. So I think there is scope of improving or finding new pathways and new ways to target even the beta receptor system.
You make a very good point about, all these drugs that are used in the clinic, and as any drug, whenever you go to the pharmacy and you get any prescriptions, there's a whole list of potential side effects. So I think going back and looking at these compounds and studying them better, understanding their effects on a cellular level, could help modify them and reduce that list of potential side effects that these compounds have, which is really important.
Agreed. I think that's a very good point. Yes.
I really love that. So you mentioned that, the beta-adrenergic receptor is one of your favorites. Any other favorite receptors that you would like to share with us?
Yeah, so I think another receptor that I just mentioned is the glucagon receptor. That seems to, anything that has link with ubiquitination and deubiquitination and shows biology, that's my favorite. So, but we have worked with a bunch of receptors. So I think I like some aspects of each receptor. So I have many favorites.
So let me ask you this: any favorite ubiquitinases or deubiquitinases?
Oh yeah, that's a good question. So right now, I think my favorite are the two drugs. That is, they are highly homologous, USP20 and USP33, and they seem to have a role in regulating at least several GPCRs. So I think those are my top candidates at the moment, my favorite ones to pursue. And that's where we started looking at the role of deubiquitination. And I think they are still my favorite dubs.
Are there any disease areas other than the cardiovascular field where ubiquitination of GPCRs or any potential targets could be important in understanding?
And so, in general, if you think about the role of ubiquitination, it could fit into regulating any GPCR, but I don't think much has been done in terms of looking at a physiological role for ubiquitination. And that's how when I started my own lab, one of my major questions was, can I find a relevance of receptor or arrestin ubiquitination in any physiological setting? Or so initially we, it was kind of challenging because you're not equipped with everything to study everything that you want to do. So I started with looking at MDM2 knockout mice, and I thought I would see some effect that takes me to beta-arrestins. And we have this paper that we published a couple of years ago, but it took a long time to get that paper done, where we actually were able to show that MDM2 is indeed important for regulating the beta receptor signaling in the heart, but it's actually because of its role in regulating GRK2 levels and not beta-arrestin levels. And if you think about this, the phosphorylation is more proximal to the receptor because it has to happen, and then you get arrestin recruitment and regulation of signaling through arrestins. And MDM2 can ubiquitinate not only arrestin, but it can also ubiquitinate GRK2. And that seems to be more important in terms of the beta-1 receptor desensitization in the heart. So when we finished the MDM2 story, we started looking at my focus when I was more interested in looking at deubiquitination. And we made the USP20 knockout mice, and we also have that paper showing that it does regulate the beta-1 receptor, and that's what I was talking about. It has a difference in terms of interaction with the two subtypes. So right now, I think we are more focused on developing that further. And hopefully our new knock-in mice can tell us whether the deubiquitination and ubiquitination play a role in cardiovascular signaling and biology. So we'll see. There might be other receptors regulated by USP20 and USP, but as far as I've seen, I think we have tested a few more receptors that are potentially expressed in the heart. And it looks like so far the only receptor that we can see a clear effect are the beta-1 and the beta-2. So we'll have to see.
That's so interesting. That got me thinking about, do you know if there are any human mutations in ubiquitinases that induce any diseases or deubiquitinases as well? I wonder, because we know that there's mutations in GPCRs that are linked to diseases, G proteins. I wonder if that's something known.
Yeah. So I think this is something that I don't think there is I can answer your question now because I don't remember seeing there could be some mutants of deubiquitinases, and they could have a role in regulating the GPCRs, but I think they are sort of maybe in parallel universe right now.
Yeah.
Somebody has to connect them.
Yeah.
Honestly, I don't know.
Okay.
I don't know, but that's something that's always at the back of my thoughts and my mind. But there are mutants that are linked with certain diseases, even in the DUBs, E3 ligases. There are diseases that are we know that the ubiquitin cascade is dysregulated, but whether it involves GPCR signaling is something that we have to figure out.
Yeah, that's why I was wondering, because it got me thinking about G proteins and mutation in G proteins and GPCRs and their link to cancer and immuno-oncology. And the link has been now established, but it took a long time for immunologists and people in the cancer field to actually recognize that there's GPCRs and G proteins that are driving the disease, or at least implicated in cancer.
Interesting. Yes.
It's I was just wondering. So we talked a little bit about the different tools that you have in the labs. You have the mouse models. You also have the ability to look at ubiquitination by pulling down the receptor. Do you think there? No, let me rephrase that. What kind of tools and initiatives we could be developing better to speed up, better understanding the signaling of GPCRs through ubiquitination?
So let me think for a minute here.
I'm just wondering if there is any way of, developing assays that would be more high throughput, or, allow you to screen, and I'm going to be very optimistic, all the known GPCRs, including the orphan GPCRs, and see if, any binders would alternate the ubiquitination of the receptors.
Yeah. So definitely, there are ways to, you can have ideas, but I don't know whether they will work. We could develop high throughput screens, but ultimately, I think to have some sort of a success, a major step, a leap in this area would involve sort of a multi-disciplinary approach. Mm-hmm. So you need different types of assays, different types of collaborations, and all coming together and addressing the question. There could be one step in a pathway that requires ubiquitination, but there could be multiple steps that require phosphorylation. Yeah. There could be multiple steps that we need to understand to put the pieces together. And I don't think one particular assay can do that. We may need somebody to tell us about the structure. We may need some people to address in terms of the signal transduction, in terms of ubiquitination. So I think if you kind of if you're able to link all these different findings, and if it makes sense, I think that is what is needed for us to make a progress towards better drugs. Do I make sense?
Yes, definitely. I think, and I totally agree with you on that. I think one great thing about science today and being all connected, although hit by COVID since last year, is the ability to connect like we're doing today, and also to bring together scientists from different fields, with different specialties. I think it's good to have a mouse model. It's good to have a structure. It's good to be able to do cryo-EM. But if you don't put all of these tools together, then I think we're all kind of working in our little corner. We're publishing papers, but in order to advance drug discovery, definitely.
Yeah, and of course it helps to have funding. I mean, it's not related to the question, but I'm just bringing up here. But that alone is not important. I think to find collaborations and to work from different disciplines. Yeah. And also some of the aspects that I want to mention that that are useful are core facilities. Like we have very nice core facilities here to help us to do some of the complex techniques which not every lab knows how to do them. And for example, cardiovascular physiology, Dr. Howard Rockman's lab and his physiology core have helped us immensely to do all our experiments. So, and then one of the tools that I don't know if we will ever be able to cross that hurdle is having good antibodies and really specific antibodies for different GPCRs. And so that's a major hurdle I think for many of us. And so those are the things I think in terms of developing new tools. And I think that's something that we always want, good antibody. And
yes, those are very difficult. And that reminds me of the story you were mentioning where you went around in the lab and with other labs and looking for flag tag GPCRs because there's no good antibodies. And a tag can be very useful, but having a specific antibody for a specific GPCR that works every time you're doing the experiment is definitely something very much needed.
Yeah.
So before we move on to the next question, before we hit record, we were talking about meetings, about conferences, and you mentioned that you're co-organizing a conference coming up soon. I would love to hear more about it.
Yes, I'm very excited that we will be. I'm a co-organizer for this upcoming FASEP meeting, which is focused on GRKs, G protein receptor kinases, G protein double receptor kinases, and arrestins. And I think this was actually supposed to have been held in 2020, and of course, like all other conferences, it got postponed. But anyway, we are hopeful that the date is now set. It is in August of 2022, hopefully. And it's going to be in Florida. So, but we are sort of working out the details. We have two organizers from the US and two from Europe. So my co-organizer in the US is Adriano Marchesi, and also Martha Sommer and Karsten Hoffman from Europe. So we hope to bring together all, top scientists, and also we want all the young scientists who are listening to this and who would like to attend. We would encourage them to submit an abstract because we want to make this a meeting more for the junior trainees, and we want to select as many talks from the submitted abstracts as possible, and we want to see them present their findings and interact. Hopefully, this is going to be an in-person meeting.
We hope so. I think
I'm keeping my fingers crossed. And we welcome any support from any listeners who are interested to support this meeting. We welcome any support also. And we also please keep watching the website for FASEP conferences, and I'm sure it'll have more details coming up in the coming months.
That's fantastic, and I'm happy to take a look also at the website and share it in the Dr. GPCR newsletter on a regular basis. I love the idea, and you made a great point about junior scientists. I love the idea of providing a platform to present and give a talk at a conference. A lot of conferences have very packed programs, and oftentimes there isn't enough time left in the program for junior scientists to give them the opportunity to actually give a talk, which is as much as writing a paper and publishing a paper part of the training.
Yeah, yes.
I love that. I love
agree
that.
With
That's
totally
great.
Agree with you.
That's something that I missed, and it was always very difficult because you would submit an abstract and there were maybe 2 spots out of a 2 or 3 day conference, and there was just only 2 people who had the opportunity to present their work. But I think encouraging young scientists to not only submit their abstract, but also give a talk is just it's just a great experience and a great way to expose their science to a larger audience. Speaking of junior scientists, any advice that you would have to share with junior scientists who want to contribute to the field?
Yes, I think so. One of the most important things for junior scientists, in my opinion, from my own experience, is that you need to find something that really incites passion. Whatever you do, if you're not really passionate about it, I think you can get only so much out of it. And definitely basic research, especially, and right now, even at any time, is not meant for the faint of heart. I mean, it needs focus, it needs dedication, it needs tenacity, it needs a certain time commitment. And it's also important that you find a support group. You're thinking about only the PI, but you should probably look at having a support group of mentors. For example, if you are a junior person, you have a senior student, senior postdoc, you have to make a connection to have an immediate mentor. And then, of course, you have a PI who is a mentor. And but ultimately, I think it depends on the mentee to make the most use out of it. So you want to get, as a young scientist, first you figure out, okay, you're passionate about this, you want to understand this, you do the experiments, but, at the regular time points, you should organize your thoughts and your experiments, and you should discuss it with your mentor or your mentors to figure out, okay, what has worked, if it has worked, and are you making progress? And if you're not making progress, why are you not making the progress? And I think your mentor should be able to guide you at that point and not suppress you. I mean, if you have to change your project, you should do that. Maybe this is sort of a dead end thing. So, and the other thing is, I think the junior scientists have to not only find, and any scientist has to find time not to just do your research and be in your well or in your pond, but you should look around. You should read as many papers, especially in the GPCR field. And I know, every day so many publications pop up, and it's very difficult to keep up, but you should try your best to not only read papers in your own research area, but also I think sometimes I found ideas or I found some insights, even reading papers that are not related to what I'm doing. Mm-hmm. And I cannot remember the exact context, but I think it's important to have a broader sense of your, reading portfolio. And of course, the most important thing is to have a supportive environment for the success of scientists. So I think the other thing is, apart from once you are familiar with your research and other things, and you have to start networking. And this networking is really important You can learn new things. You can, if there is sort of a forum for networking, I think that is very good for young scientists. This will help them not only to become more creative, but also, to sort of start presenting your ideas formally and informally. That's also an important thing. So those are the things that, if you make a checklist, those are some of the important things that you may have to do to sort of successfully contribute to the field.
I love that. Thank you. You touched on a very interesting and important point, which is networking. And I think it's important to go lift your head up. Don't keep your head down on your bench, and go out there and actually talk to people, even though they may not be in your field. But comparing notes and sharing ideas can be very conductive to new discoveries. And you never know who you'll meet, or who will make an introduction, and who will open up doors for you. But at the same time, you may be also the person, you can help someone else succeed. And I think that's very important in science, but in general, I think as humans, but definitely in science.
Yes, exactly.
Super.
I couldn't agree more.
And it's interesting because when I was a PhD student, I did have a lot of friends who were also doing a PhD in immunology and other fields, but for some reason, I spent, and I learned this the hard way, I spent a lot of time trying to solve problems on my own, until one day I realized, actually, if you ask for help, most of the time people are happy to help. Most of the time, people are happy to answer your questions, and most of the time, people respond to your emails. So, if I had advice to my younger self, is just go out and just ask. The worst that can happen is that you don't hear back, or you're told no. But then there's other people you can ask for input. And I don't know anyone in the GPCR field who would say, "Well, I don't want to talk to you." Or I don't want to help. Even if they have 5 minutes, they will definitely give you that.
Yes. From my experience, what you said is correct. I mean, I have approached different people for different things, and I've always received help, advice. Yeah. I mean, sometimes on some occasions, either they are too busy, and then you can always find somebody who has a little bit time for you.
Exactly. And I think the important thing is to go out, reach out to people, and respect people's time as well. And if you get help, give help as well, because I think it goes both ways.
Yes, exactly.
So before I let you go, I asked this from everyone. So what are the top 1 to 3 aha moments that define your career?
I know you didn't ask, but I really want to address this, because this is one of the important things for me.
Of course.
One of the points that I didn't get to mention is, I believe that having diverse viewpoints from students, trainees, colleagues is very critical for making progress.
Yeah.
And I believe that creativity and innovation has no barriers, should not see any barriers, and everyone, irrespective of where they come from, what they are, I think they should be encouraged, and they should be able to contribute and be heard. And this is, I'm a firm believer of this. So I just wanted to mention that.
I think it's important. I love that. I think it's important. I think it's important to be open to other opinions. Yes. And as long as it's done, respectfully, and it's done in a smart way. And I think, data drives the science, and I think data should drive the field forward, independently of who did the experiment. Or data is the data, and I think numbers are a good way to rely on it. Agreed.
Yeah, and you were asking me about the aha moments I have had. Yeah, I think I can think of several. But every time I see some data that says something, whether it is just a gel where we are cloning something, it always excites me. I'm very easy to feel happy about something. It may not mean publishable data, but to me, every step is important and enjoyable. But if I think back, what were the moments that really sort of I felt, I don't know. So the first thing I can recall is the big relief I had when I saw the QPS protein band, which was like sharp and glowing at me instead of sort of a smear at the end of the gel. And the second thing in my PhD, I can still remember when I picked up the facsimile or the fax that used to come to our printer from this sequencing facility that was somewhere in Oklahoma City, and it had the sequence, the protein sequence, but that was not the important thing. The important thing was a comment that I can never forget from the sequence facility director, that he said he has never seen this sort of a protein sequence, and all the peaks are so clean and no background, and there were like 43 amino acids he could easily call. So the total length of the protein is 100, and we could get a sequence of 43 amino acids. So I can never forget that, reading that comment from him. And then, of course, one of the very exciting moments that I can never forget is when I did the confocal imaging of the beta-arrestin ubiquitin chimera, when as a postdoc, and then it would light up the endosomes, and which was unexpected. And also when I made the opposite, when I took the wild type and mutated 2 of the residues, and the lysines 11 and 12, and now it lost the ability to get into endosomes with certain receptors. And this brings me back to what I was telling you about the ubiquitin code, because there could be sites that are specifically ubiquitinated in certain GPCR pathways, and we don't know them. And it is one of the things that I always think about. So I think those I can never forget those the feeling I had when I did those experiments. So those are the things. But in general, in my own lab, I am always looking at experiments on a daily basis. Sometimes I even look at experiments that I insist that they show me experiments that failed, because I think there is something to learn from those experiments. Either we are asking the wrong questions, or we are doing the wrong experiment. So I insist that we discuss failed experiments also, and not just the data that we got, So that's one of the things that I like doing.
I love that you mentioned, looking at the failed experiments, because as you mentioned, it's very as it can be as informative as successful experiments, and it can, put in perspective questions, hypotheses, and also give you ideas on which way to go, or sometimes which way not to go.
Yes. And it could be because of a reagent. I mean, I can remember at one point, someone was doing mutagenesis, and then it turned out to be that the oligo was made wrong. And no matter how many times you did the mutagenesis, it wouldn't work. And all we had to do was resynthesize the oligo.
Yeah. And sometimes
you
it's
have
also
to
simple.
talk. I mean, yes. I think the students have to, that's the point of discussing with your immediate mentor and getting, not just keeping it aside. It's not working. And, there's always a reason when things don't work, at least the things that are supposed to work.
Exactly. I think it's very important also to always ask the question, why? Did it work? Okay. Why? It didn't work, then okay, why? And how can we improve on that?
Yeah.
All right. Before I let you go, I also ask always this. I always ask my guests this: if and when you have job openings in your group, where can people find you?
In general, I use the Duke supported websites usually, but anybody who is interested in can email me, and I think I know that your website also has the opportunity to advertise. So Yeah.
We're working on automating it a little bit better, but yes, there is opportunity. I think.
I mean, anybody who is intrigued by ubiquitination and ubiquitin-dependent regulation, please contact me. I'm happy to discuss things with you.
Perfect. That's perfect. I will, once the episode comes out, your contact information and link to the Duke website will definitely be on the webpage, and people will be able to find you.
Thank you.
Of course. And with this, Sudha, thank you so much for your time. I really enjoyed our conversation.
I did too. And I cannot just believe that we have talked for almost an hour, because when I thought me talking, it could be probably only 15 minutes because...
Nope, that's it. So I'm going to hit...
that's because of your influence. And I have to tell you that I really enjoyed talking with you. And I think I'm so happy that you're doing what you're doing. And I want to wish you the very best, and keep doing this. And I will keep in touch with you.
Thank you so much. I really appreciate it. I'm going to hit stop. Please don't go anywhere. All right. Thanks for being here. We'll talk to you soon.
Thank you for joining us for this Dr. GPCR podcast episode. I'd like to thank our guest, as well as our team members, Attila Forrest, Shivani Sajdev, Alexa Druan, and Ines Pinero. We look forward to seeing you live at the Dr. GPCR Summit in September. Don't forget to visit drgpcr.com/summit. Please subscribe to the Dr. GPCR newsletter, find us on YouTube, and if you like our podcast, leave us a 5-star review on Apple Podcast. You can also leave us testimonials at drgpcr.com/testimonials. Another way to support us is to share your favorite Dr. GPCR program with your network and colleagues. Email us with any questions or suggestions at hello@drgpcr.com. Until next time, stay safe.